Okay, I'm going to start to review anatomy and physiology. 2. This is the value 212 subject. So, body movements. There are 600 muscles and 206 bones. Bones and muscles function together as levers. The lever components, we have functioning parts, the rod, it's called the stick, pivot, fulcrum, force, effort, one weight or load. So 3 kinds of levers or types of levers. We have the first class, second class, and third class. The first class, the sequence is weight or load, and then pivot, and then force. The second class lever, the sequence is pivot first, and then the weight, and then the force. For the third class lever, the sequence is pivot first and weight. So there are types of muscle tissue. We have skeletal, cardiac, and smooth. The smooth is the internal organs or blood vessels. Skeletal muscle is the one that's attached to the bones, cylindrical fibers. It has multinucleate. It's voluntary and strategized. Then we have the cardiac muscle, which is found in myocardium. Yes, the heart wall has branched fibers, 1 to 2 nuclei per cell. It's involuntary, stratiated, and intercalated disc. Then we have the smooth muscle. It's found the walls of an organs or blood vessels. It's the spindle fibers. Has 1 nucleus per cell, involuntary and non-stratiated. The muscular system consists of over 600 muscles. Muscles that move bones, or it's called locomotions. Muscles, or MM, are organs. We have skeletal muscle and connective tissue called fascia. There are types of fascia. We have superficial, which is at the bottom layer of skin. It is deep to skin, adipose tissue, and loose connective tissue. And then we have the deep fascia, fibrous connective tissue, lines body walls, extremities, holds muscle together. And then we have the extension of deep fascia. Into muscle, we have epimysium. Epi means above. This is around whole muscle. And perimysium, around fascicles, bundle of muscle cells. Endomysium, around fibers. So beyond muscle, we have tendons, muscle to bone. Aponeurosis, connect muscles. Muscle types: prime mover against or prime mover, agonist, muscle that causes action, moves. We have synergist, not causing the action but helps stabilize the joints, which helps movement. Antagonist opposes the movement, example, the biceps. We have the muscle parts, origin, less mobile end, insertion, more mobile end, belly, body, contractile. Muscle nomenclature: origin or insertion, sternocleidomastoid. Number of heads of origin, biceps, brachii, quadriceps femoris. We have the size, pectoralis major, shape, deltoid, action, extension digitorum, fiber direction, external abdominal oblique, location, rectus femoris. Okay, and we have the skeletal muscle histology. Fiber or muscle cell, long, thin, cylinder. Sarcolemma, cell membrane. Transverse tubules, extensions of membrane. Sarcoplasm, cytoplasm, it's the fluid inside the cell. Inside cytoplasm, we have organelles, or inside cytoplasm, or organelles in sarcoplasm. We have ribosomes, lysosomes, and many more. We have many nuclei, contain many mitochondria, energy. Sarcoplasmic reticulum, this stores calcium. Terminal cisternae near T tubules. Myofibrils or fibers contain myofilaments. There are two types of myofilaments. We have thick and thin. Thick filament made of protein called myosin. It's a twisted golf club shape. Head has ATP binding site. For the thin filaments, we have three proteins. We have the actin, treponin, tropomyosin. Treponin, tropomyosin helix. Actin, it's the kidney bean shape, has myosin binding site. When muscle is at rest, troponin, tropomyosin complex coveals myosin, it's a binding site. Contraction, the myosin pulls actin. Sarcomere, flash unit or unit of contraction of muscle. Structural unit of contraction, we have region between Z lines. It causes striations. Components, A band, I band, H zone, M line. A band is a wide dark stripe, width of thick filament. Sorry, here. I'm gonna review that later on. Okay, for the I band, we have the light stripe, region between thick filaments, only thin filaments. Edge zone, center of A band, only thick filaments. It's lighter. M line, proteins connect thick filaments, center of edge zone. Z line, center of I band. Thin filaments attach. The neuromuscular junction, neuronerve, contact point between nerve fiber or cell and muscle. The parts of neuromuscular junction, somatic motor neuron, synapse, nerve comes from close to the muscle, muscle fiber, pliers. So somatic motor neuron, somatic, it's voluntary control. Motor signals travel from central nervous system to effector. We have telodendria, branches each to the muscle fiber. So each one gets to individual muscle cell. The motor unit refers to single somatic motor neuron and all muscle fibers innervated by neuron. Synaptic ends bulbs. This is the swellings at ends of telodendria. It contains synaptic vesicles, carry neurotransmitter, and then after that, acetylcholine in skeletal muscle. Synapse, area or gap between nerve muscle. Acetylcholine carries signal across. Muscle fiber. Motor end plate is the near neuron, mainly found in mitochondria. It's folded sarcolemma. For the muscle contraction, alright, so energy sources for muscle contraction. We have phosphagen system, glycogen, it's a lactic acid system, cellular respiration. So phosphagen system, creatine phosphate, and then creatine plus the phosphate plus energy. ADP plus energy equals ATP. Enough energy for 15 to 30 seconds of muscle contraction. For the glycogen lactic acid system, this is anaerobic. It doesn't need oxygen, glycogen, then goes to glucose to do glycolysis, plus lactic acid plus ATP. Enough energy for 40 seconds of muscle contraction. This builds up lactic acid. So lactic acid lowers pH, soreness, diffuses of muscle cell carried to liver. Lactic acid convert glucose, ATP broken down ADP. So cellular respiration, this is aerobic, that means it needs oxygen. Carbohydrates, other organic compounds used to get ATP, provides energy after 30 seconds of muscle contraction. Two sources of oxygen, diffuse from blood, hemoglobin, RBC or erythrocyte, these are red blood cells. Release from myoglobin. Myoglobin is the protein inside muscle cells. So this one stores oxygen when muscle contracted. When contract muscle, you build up oxygen debt. For oxygen debt, we have energy used during contraction. The sources of debt, we have ATP for lactic acid, turns to glucose, stored ATP, and stored creatine phosphate. This is being repaid by rapid or deep breathing, oxidized glucose, or this is the ATP. Strength of muscle contraction depends on individual muscle fiber or muscle cell, all or none. So muscle organ contracts graded strength, not all or none. Factors affecting contraction or are ATP plus calcium, but we have fiber length, load weight, metabolic conditions, number of motor units, recruitment bringing in more motor units, stronger. And we have the contraction types. We have tonic, isotonic, isometric, twitch, tetanus, fibrillation, convulsion, trap, rigor mortis. And we have tonic contraction. The tonic contraction is a continuous partial contraction. Muscle tone, alternate motor units regulated by muscle spindles. It monitors muscle length, triggers stretch reflexes. For the Golgi tendon organs at muscle tendon junction, monitor tension during muscle contraction. The isotonic contraction, muscle tension stays the same, and the muscle shortens, and it has normal movement. The isometric contraction, the tension increases, and muscle length stays the same. So I'm just gonna continue studying next time, but that's how far it gets from my study. So these are the first class lever. This is the seesaw, like scissors. So first class, this muscular system, rod, forearm, pivot, and elbow, and force and triceps. And we have the wheelbarrow, trace body and toes, the rod, bones of live foot, the weight is the body, the pivot is ball of foot, and force calf muscles. We have the tweezers, the tweezers, the flexion of arm. We have the rod is the forearm bones, the weight is the hand, and pivot is the elbow, force is the biceps. And we have the type of muscles. We have skeletal muscle, cardiac muscle, and smooth muscle. Hard and smooth. Let's see, these are extensions of deep fascia. Let's see. Okay, so we have the tendon, epimysium, endomysium. These are between fibers. We have the bone, perimysium. The fascicle is wrapped by perimysium. These are the blood, endomysium, muscle fiber, and then we have this one over here, closer one, in the middle of fascicle. We have endomysium, perimysium, and the red one, blood vessel. So beyond muscle, we have the cranial aponeurosis, is temporal, occipitalis, masseter, sternocleidomastoid, trapezoid. We have the frontals, orbicularis, oculi, zygomaticus, orbicularis oris, platysma. We have rectus femoris, vastus intermedius, vastus lateralis, vastus medialis, patella. We have pectoralis major, and we have the deltoid, and we have the serratus anterior. It's the abs, the external abdominal oblique, rectus abdominis. It's the abdomen, but in the middle, located along the front of the abdomen. And we have transverse abdominis, located under the oblique, deepest of the abdominal muscles and wraps internal abdominal under the external oblique. Okay, anyway, this is the cell. This is my filaments. So sarcoplasmic reticulum, mitochondria, T-tubule, sarcolemma, thick myofilament, thin myofilament. Nuclei, fiber, sarcolemma, mitochondrion, myofibril. Dark is the A band, light is the I band. I have microfilaments in here. Part of skeletal muscle fiber or cell, sarcolemma. I band, A band, C disc, M line. Okay, and we have sarcolemma, triad, mitochondria, myofibrils. We have tubules of sarcoplasmic reticulum, terminal cisterna of the sarcoplasmic reticulum, T-tubule. Okay, let's see this one. Okay, sarcolemma, mitochondrion. Dark is the A band, light is the I band. It's the nucleus, C disc, H zone, C disc, I band, A band, I band, sarcomere, M line. And then we have the thick filaments. This is the ATP binding sites. Those are the heads. And yeah, myosin head. And we have the thin. We have troponin complex, tropomyosin, and actin. So troponin is the white, tropomyosin is the stripe, and the actin is the bean shape. So longitudinal section of filaments within one segment of myofibril. And we have sarcomere, C disc, H zone, C disc, I band, A band, I band, M line. So the M line is this in the middle, C disc, thin filament, okay, and then we have sarcomere, Z line and M line. So H zone, is that, A zone is that, I band, neuromuscular junction, somatic motor neuron, synaptic bulb. Myelinated axon of motor neuron, action potential. We have axonal terminal of mitochondrion, synaptic cleft, T-tubule, junction folds of the sarcolemma at motor end plate, part of myofibril, synaptic cleft, axon terminal, acolyte molecules, acetic acid, choline, acetylcholinesterase, binding of alkaline. The receptor opens sodium channel. And we have the somatic motor neuron. See what it looks like. Spinal cord, motor neuron, cell body, muscle fibers, and terminals of neural junction. Those are the muscle fibers. These are the one inside the muscle. These muscle fibers and branching axon to motor unit. Sodium and potassium stimulus. So for the electrical conditions of a resting sarcolemma or polarized, the outside phase is positive while the inside phase is negative. The predominant extracellular ion is sodium, so positive, negative. That one is at resting. The predominant intercellular ion is potassium. The sarcolemma is relatively impermeable to both ions. And for depolarization and generation of the action potential, production of an end plate potential at the motor end plate causes adjacent areas of the sarcolemma to become permeable to sodium. Voltage-gated sodium channels open. As sodium ions diffuse rapidly into the cell, the resting potential is decreased. Depolarization occurs if the stimulus is strong enough and action potential is initiated. So it's over there. So we have positive, supposed to be all the positive is outside, and then it just changed from some of the negative went out and the positive went in. It's 5. We have 6. This is more it's the same. Like all the positive is outside, some of the negative just went in. So step 2 is propagation of the action potential. The positive charge inside the initial patch of the sarcolemma changes the permeability of an adjacent agitated sodium channels there. Consequently, the membrane potential in that region decreases and depolarization occurs there as well. Thus, the action potential travels rapidly over the entire sarcolemma. Let's see that. And for step 3, repolarization. Immediately after the depolarization wave passes, the sarcolemma's permeability changes once again. Sodium channels close and potassium channels open, allowing potassium to diffuse from the cell. This restores the electrical conditions of the resting polarized state. Repolarization occurs in the same direction as depolarization and must occur before the muscle fiber can be stimulated again. The ionic concentrations of the resting state are restored later by the sodium-potassium pump. So that's the sample. There's the potassium going out, the positive ions going out, and the negative ions going in. Okay. And we have the axonal terminal. This is the synaptic cleft. Neurotransmitter releases diffuses across the synaptic cleft and attaches to acetylcholine receptors on the sarcolemma, ACH. The sarcolemma T-tubule. Action potential generated is propagated along the sarcolemma and down the T-tubules. Have all calcium outside. 2. Action potential triggers calcium release from terminal cisternet of SR. Cisternet. And 3. Calcium ions binds to troponin. Troponin changes shape, removing the blocking action of tropomyosin. Actin active sites exposed. And then 4, we have the contraction. Myosin cross bridges alternately attached to actin and detach, pulling the actin filaments toward the center of the sarcomere. Release of energy by ATP. Hydrolysis powers the cycling process. We have 5. Remove of calcium plus active transport into the SR. Enter the action potential ends. Tropomyosin blockage restored blocking actin. Active site and then contraction ends and muscle fiber relaxes. So removal of calcium plus active transport into the SR. Enter the action potential ends. So actin, troponin, myosin, tropomyosin. That's myosin head. Troponin complex. It's the white one. Calcium ions. It's the head. That's myosin head. Myosin cross bridge attaches to the actin. And then there's ADP, phosphate, inorganic phosphate release. ADP working stroke. The myosin head pivots and binds as it pulls some actin filaments sliding toward the M line. As a new ATP attaches to the myosin head, the cross bridge detaches. And as ATP is split into ADP and phosphate, packing off the myosin head occurs. Take filament, ATP, hydration. Then filaments. And this is the same. We have the H line, Z line, I band, A band. Whole thing is A band. So sources of energy for muscle contraction. We have CP, ADP, creatine, ATP. Direct phosphorylation coupled reaction of creatine phosphate and ADP. Energy source, creatine phosphate. Oxygen use, none. Products, 1 ATP per creatine phosphate. Creatine duration of energy reduction is 15 seconds. Then after that, glucose from glycogen breakdown of delivered from blood. Glycolysis in cytosol. Pyruvic acid, lactic acid. 2 ATP again released to blood. Their anaerobic mechanism, glycolysis and lactic acid formation. The energy source is glucose, oxygen use, none. Products, 2 ATP per glucose, lactic acid. Duration of energy provided 30 to 60 seconds. And the glucose from glycogen breakdown or delivered from blood. Pyruvic acid, fatty acid, aerobic respiration in mitochondria. Water, carbon dioxide, ATP, amino acid, net gain, or glucose. So aerobic mechanism, aerobic cellular respiration. Energy source is glucose, pyruvic acid, free fatty acids from adipose tissue, amino acids from protein catabolism. Oxygen use required. Products, 38 ATP per glucose, carbon dioxide, and hydrogen. Duration of energy provision, hours. Then we have the factors affecting contraction. So optimal sarcomere operating length. You're still seeing the H line, A line, Z line, there, I band, A band. Anyway, so single action potential initiated. There is some example for light load, intermediate, and heavy load. Then we have the another sample in here. Speed of muscle, number of motor units, recruitment. Maximal stimulus, tension, motor unit 1 recruited small fibers, motor 2 units, medium fibers, motor unit 3 recruited large fibers. More, and then time. So tonic contraction, this is the muscle spindles. Efferent motor fiber to spindle, secondary sensory endings, primary sensory endings, muscle spindle, connective tissue capsule, extrafusal muscle fiber, infrafusal muscle fibers, efferent motor fiber to extrafusal muscle fibers. Then we have this capsule, the Golgi tendon organ, sensory fiber, tendon, Golgi tendon organs attach muscle to bone. So isotonic contraction. We have tendon, muscle contracts, isotonic contraction, shortens, tension develop, muscle length, percent of resting length. There's the tension develop, amount of resistance, muscle relaxes, muscle stimulus, muscle resting length over the muscle length, percent over resting length. Oh, and then and this the same isometric contraction. Muscle contracts, isometric contraction, develop tension, develop muscle length, amount of resistance, peak tension, develop muscle relaxes, muscle stimulus, resting length. So, and then muscle twitch, 3 phases. Latent period, period of contraction, period of relaxation. So latent, extraocular muscle, gastrocnemius soleus. Yeah. Then we have tetanic contraction, incomplete to complete. There we go, that's the tension. And we have this. This is the wavelength. It's post-mortem interval. Let's see what else is going on in here. I think that's it for now. Let me see. We get another one. We have some other notes in here. Hello.